Hey everyone, Professor Bob Long here doing your anatomy and physiology lectures. As everyone knows, we are in the coronavirus or COVID-19 lockdown, so we're having to transition all our face-to-face courses to an online format. So I'm doing this series of videos so my students can keep caught up with materials and we can finish the semester on time. If you're in my class, this lecture is going to be muscle lecture number 7, I believe. It'll be in the title of the lecture. And this is going to be over muscle performance, recruitment, wave summation, and a few other concepts like that. If you're following along in my note set, forgive me for a second for stepping out of screen, but I'm going to do that a few times. We're actually starting in my note set, I believe on page 57. Yes, it's on page 57, and we're going to run through the next few pages. Those of you that are in my class, follow along in the note set, download the worksheets, and try to fill that in. Those of you that are not in my class, if you're watching this and you learn something from it, great. I hope it helps, and I hope you'll have as much fun as I do. One last detail, I am filming using my cell phone and my laptop. The laptop allows me to see what I'm doing. The cell phone does not. So if I step forward, sometimes it's going to cut my head off or I'll step out of screen. I apologize. These are being done kind of impromptu with a minimum of equipment, the best way that I can just for now. Ultimately, what's important is what's going to be on the board, and that's what you need to know. Also, I'm having some severe allergies the past few days. My mesquite tree is trying to mate with me, and the pollen's getting to me. I may have to take a drink occasionally just to keep my throat going, and I have a little cough. I don't have the virus. I don't have any temperature or any other symptoms. I'm just fine. Anyway, so if you guys want to follow along here, and what we're going to be talking about is muscle performance. How do muscles actually perform their activities? In order to do this, I need to back up a second and go over a little bit of what we've covered in a previous lecture. If I were to draw an individual muscle cell here, so here's my single muscle cell with its sarcolemma. I'm not drawing the fascicle and the muscle fiber and all of that. And as you guys know, on the outside of the muscle cell, there's a connective tissue covering called the endomysium. Now, if I look at the cell membrane, I see these little holes every now and then, and these are holes that would run through the muscle cell. They are invaginations that run all the way through. Kind of like your body has the external surface, your skin, that kind of invaginates in and forms the oral cavity, runs through the digestive tract, and comes out the other end. It's all the same opening. If I drop this pen in one end, eventually it will come out the other end. And the same thing happens for the cell. If I drop this, it would fall all the way through. Now, I'll come back to that in a second, but you should know that each one of those structures is called a T-tubule or a transverse tubule. So we see T-tubules on the surface of our muscle cell, okay? Now, if I take out some of the myofibrils. A muscle cell is a bundle of myofibrils, those long structures that are made up of proteins called myofilaments, actin and myosin, or the thick and thin filament. If I pull one myofibril out, and I start to look at the ends or look along its length, I'll see my actin and myosin filaments like this going throughout its length, okay? Sorry, it's a little messy. So we can see how my actin and myosin filaments are making my sarcomeres, and they're repeating subunits on here. There's my Z lines and my actin or thin filaments. There's my M line and myosin. And you'll see these subunits repeating, so on and so forth ad nauseum. Now, one of the things we talked about in the last video was that the endoplasmic reticulum of a muscle cell, called the sarcoplasmic reticulum, is wrapped around this myofibril, and the terminal cisterna sit right around the zones of overlap. And then we see all these other cisterna going through here. And if I look through the gaps between the tubules, I can see the actin and myosin underneath. And I'm not going to draw this anatomically correct for now. This is just to get the concept across. But if I have another section of sarcoplasmic reticulum here, interrupting each section of sarcoplasmic reticulum would be a T-tubule. Now, mind you, this is one myofibril with the sarcoplasmic reticulum wrapped around it with the T-tubules interrupting it. Every myofibril is the exact same setup. Now, the muscle cell membrane, the sarcolemma, would extend down here. And let's say, for example, I have a motor neuron synapsing right here with its synaptic knob around a motor end plate, going to release neurotransmitter acetylcholine to bind to acetylcholine receptors. As we covered in the last lecture, as soon as acetylcholine binds to the receptors, that starts an action potential spreading down the sarcolemma in both directions, this way and this way, because of the number and the location of all these ion channels. As the sodium ion channels allow the action potential to spread, it will spread down the sarcolemma and down every T tubule, shocking 100% of the sarcoplasmic reticulum and 100% of the myofibrils, causing 100% of the sarcomeres to contract. So when one muscle cell contracts, it contracts with 100% of its tension. Every myofibril inside the muscle cell, every sarcomere in every myofibril will contract. And that gives the same amount of tension for one muscle cell, so to speak. We call this the all or none principle. In the all or none principle, essentially what it says is, if one muscle cell contracts, it contracts with all of its tension, 100%, or none, 0%. It's either all or none for one muscle cell. Now, what's counterintuitive about that is, if a muscle cell contracts with 100% of its tension, how do I control the tension in the muscle to lift different objects? For example, let's say this black marker represents a 10-pound weight. This green marker represents a 50-pound weight. I can go and curl a 10-pound weight. I actually can't curl a 50-pound weight at this point in my life, but let's say it's a 25-pounder. I know I can curl that. So I got to be able to curl a heavy weight, turn around and grab a lighter weight, and not slap myself in the shoulder with it. I have to be able to contract it under control. How do we vary the tension in a muscle knowing the all or none principle? And it's based on two other concepts. One is called recruitment. Recruitment is increasing the number of active motor units. And we're going to define a motor unit in a minute, which is the number of muscle cells controlled by a single motor neuron. I'll get into the details of the definition. The second way that we can increase the amount of tension in a muscle is by what we call increasing the frequency of stimulation. So I'm going to go over these two concepts and explain them so that hopefully they make sense in your brain. So I'm going to start with recruitment. In order to understand recruitment, I need to understand what a motor unit is. So now, I'm going to give you a simple concept or simple idea because I'm a simple guy. I'm not all that bright. I have to use simple concepts. So let's say I take this picture here. I have a car. My car and this picture is going to represent a piece of bone. I have a long rope connected to a person who can pull on it. This is my muscle cell or a myofiber. This is the endomysium, the connective tissue. When a muscle cell pulls, it's pulling on its endomysium. Excuse me. So now, let's say I take another muscle cell and I weave their endomysia together all the way down to the bone. And I take another muscle cell and another one and another one. I have 5 muscle cells here, and all of them have their individual endomysia woven together with the endomysium of the others. Now let's say I take a few other muscle cells and they have their endomysium woven in with these. And I'm going to draw 5 of these guys, and their endomysia is going to get woven together like this. And I got to draw one more person out here. Again, this is the endomysium. But this part, where they're separated, but where all the black fibers are woven together or all the green fibers are woven together, that would be the perimysium of the fascicle. Okay? And then where all of those are woven together, that becomes the tendon at the end of the muscle. And for a little while, we call it the epimysium. And that makes the tendon at the end of the muscle. So essentially here I have 10 muscle cells. And they're broken into two groups. All of the black muscle cells are stuck to the same piece of perimysium. So this would be a fascicle, and that would be a fascicle. Now, let's say I have a neuron, and this neuron only controls these two muscle cells. It actually splits and controls more than one muscle cell. This is necessary in the human body because some of our muscles, like the femur muscles and the back muscles, the quadriceps, I should say, the back muscles, some of the larger muscles of the body can have thousands and thousands of cells. If I had to have every cell controlled by a single motor neuron, this would be my motor neuron coming out of my spinal cord like we drew in the last video. If I had to have one motor neuron for every muscle cell, then my spinal cord would have to be this big around. It'd be the size of a telephone pole or a tree trunk. And the motor cortex and the cerebellum of the brain would be huge considering all the muscle cells we have in the body. So in order to decrease the number of motor neurons and the amount of brain space and spinal cord and nerve space I have, what we have is what are called motor units. A motor unit is all of the muscle cells controlled by a single motor neuron. And in this particular motor unit, I only have two muscle cells. Let's say I have another motor neuron here, and at the very end, it's going to branch and synapse on 3 muscle cells. So this fascicle would only have 2 motor units. One motor unit has 2 muscle cells, the other one has 3 muscle cells. I didn't draw that quite right, but you get the idea. Okay? The other fascicle could have motor units as well. So you guys need to understand the definition of a motor unit. When we talk about a motor unit, it is all the muscle cells controlled by a single motor neuron. That's all that a motor unit is. Now, I just wanted to make sure that I'm catching it on my phone so that y'all can see the whole board. It's kind of why I have these lines on the board, but anyway. Now, when one motor unit contracts, so let's say, this is just an example, it's much larger than this, more numerous than all of this, but let's say that in this muscle, I have all these motor units. Okay? I got 4 motor units divided up here. Let's say only this motor neuron fires. Am I going to get 100% of the tension in the muscle? The answer is no. I get 100% of the tension out of these 2 cells. That's only 20% of the tension that the muscle can provide. So, essentially what's going to happen is, when I see something that I want to lift, my brain kind of guesses how heavy it's going to be, and then tries to fire the proper number of motor units to move the object the desired distance. For example, let me pull this chair over here. I got a chair that's pretty heavy. Now, if I want to push this chair only a few feet, then I'm going to fire a few motor units. If I fired the same number of motor units, I'm going to move the chair the same amount of time because I have two forces battling each other. Resistance of the chair due to gravity and weight and mass and friction, the frictional coefficients of the wheels and the carpet and all of that. And the force generated by my muscle, the tension. When tension overcomes resistance, the object will move. If tension is less than the resistance, the object will not move. Now, if I only generate a certain amount of tension, every time the chair will move the same distance. If I needed to move farther, I got to generate more tension. So let's say in the first movement, I contracted, say, this motor unit and that motor unit. I only needed 40% of the tension in the muscle to move the chair a short distance. If I needed to move it a much further distance, I might have to fire more motor units. We call that concept recruitment. This is the first way that we increase the tension in a muscle. There's 2 ways we can increase tension in a muscle. One is recruitment. Recruitment is an increase in the number of active motor units. My brain guesses if that number of motor units doesn't get the desired effect, then I fire more motor units or more motor units. A good example would be this. This happened to me one time. I was at an outdoor store looking for yard stuff. This giant rock was sitting there. I thought it was a real rock, and I just went to see how much it kind of weighed. I didn't realize it was a very good model, plastic model, lightweight plastic model of a rock. It looked real. And in the middle of the store, my wife and everyone standing around, I go to just see how much it weighs, and I lift a little bit, and boom, fell flat on my rear end. That's because my brain thought it was going to be so heavy, it fired more motor units than I needed. So I don't want to smack myself in the face every time I pick up a cup of water to drink. So I don't need 100% of the tension in the muscle. So when one muscle cell fires, it uses the all or none principle, 100% of its tension or not. But just because one muscle cell fires doesn't mean that's 100% of the tension in the muscle. To increase the tension in the muscle, to get the desired effect, I can do recruitment. I can increase the number of active motor units, therefore increasing the number of cells pulling. Just like if we were at the beach and somebody was stuck and we were trying to pull their car out, and let's say 2 of us weren't getting the job done, then someone else could call a few more friends and say, hey, let's come over here and I want y'all to grab onto these ropes and pull, and we could eventually move the car. Now, that's one way that we increase tension in a muscle. The second way I'm going to go over is called increasing the frequency of stimulation. What that's going to require me to do is I'm going to erase all of this. So now, let me erase all of this, and we can start with the frequency of stimulation and see how that works, okay? Now, I apologize for the cough. It gets worse the longer I talk. So let's say, for example, I could take a muscle out of an organism and attach that muscle to a few little meat hooks that have a spring down here. And when the muscle contracts, this spring will measure the tension. And I can stick that on some oscilloscope or something, and every time I have a little button here, and I have a little dial, and the dial has numbers, 1, 10, 100, or 1,000, or whatnot. I know you may not be able to see those numbers very well. It's not important. Listen to the concept. So let's say I set this button on 1, and I press the button, and that means every second I'm going to shock this muscle with an electrode 1 time per second. And the muscle will contract, and I will see the tension here. That allows us to draw what we call a myogram of a muscle. A myogram is really basically a diagram of the tension in a muscle that allows us to understand how increasing the frequency of stimulation can increase the tension in a muscle, so we can increase the force the muscle generates. So now, if I were to take this and draw it out a little bit bigger and better, it would look like this. What I have over here on this side is tension in the muscle, and I'm not going to put numbers with it. I'm not interested in the numbers. And down here I would have time in milliseconds, thousandths of a second. Again, I'm not interested in that. At 0 time and 0 tension. If I do nothing to the muscle, nothing, I don't stimulate it at all, it turns out that the tension is not at 0, it's sitting at some number a little bit higher than 0. And we call this range the muscle tone. I hope y'all can see that in the screen on my cell phone. I'm right about the edge of the visual field, so I apologize. Let me see if it's showing up. Again, I apologize for this amateur nature. Yeah, you can see it. So now, the tone is the amount of tension in a muscle while the muscle is relaxed. It never goes to 0 tension unless you're dead. Essentially, this is what's happening. Let's say we were going to have a tug of war with another class, and whoever's class moves a marker, a flag 10 feet first, gets an A for this semester and free tuition for the next semester. Well, if we were going to battle another class and I let y'all hold on to that rope, y'all can't tell me that y'all would let the rope be sitting on the ground. You would want to be pulling on that rope until there's a little bit of tension in the rope, so that the minute we start pulling, that flag starts moving. We don't want to have to take all the slack out before we get action. In an animal, that means survival. If a deer or an antelope is about to take a drink of water and an alligator or a crocodile pops out, boom, they got to jump and have action right away. So our muscles have a small amount of tension in them. They're just waiting for that real life activity, so they're not at zero. They have some tension in them called the muscle tone. Now, let's say at some point in time, I press that button, like right there. That point in time. There is a small period of time, and it is very small, but if you amplify the signal enough, you'll see it. That is called the latent period, or the latent phase of a muscle twitch. What we're about to draw is a muscle twitch. The definition of the latent period is the period of time during a muscle twitch between when we have the stimulus and we begin seeing the increase in tension. So it's a period of time after the stimulus. Let me reword that. The latent period is a period of time during a muscle twitch where after we stimulate, we do not see an increase in tension. It is a very small fraction of time, but it's there. And essentially the latent period comes from the structure of the muscle that I have erased, because when the neurotransmitter is released, or when we stimulate the muscle in any way, there's a fraction of a second for the action potential to spread down the sarcolemma, down the T-tubules, shock the sarcoplasmic reticulum into releasing calcium, calcium to bind to troponin, troponin to change shape and pull tropomyosin off the active site, and the myosin heads begin to start pulling, and we see the tension increasing. That's the latent period, is that period of time for all that biochemistry to occur. As soon as it does, we see this. This area in green is called the contraction phase. The contraction phase of a muscle twitch, or the contraction period, is that period of time after a stimulus where we see an increase in tension in the muscle. And then finally we get to the last part of it, which is called the relaxation phase. During the relaxation phase, what we see is it's the period of time during a muscle twitch where we see a decrease in tension in the muscle. This whole thing would be a muscle twitch. A muscle twitch is usually defined as a single stimulus, contraction, and relaxation period of a muscle contraction. So, you need to know the definition of muscle tone. You need to know the definition of the latent period, the contraction phase, the relaxation phase, and what a muscle twitch is. Now, what we do know is that twitches do not accomplish anything useful. If I wanted to take a drink of water, if I twitched the muscles in my biceps, I would never get the drink to my face. It would contract and relax, contract and relax, contract and relax. That doesn't accomplish anything useful. So, now, what if I set this dial to stimulate one time every second if I hold the button down? Over time, what I would see is a series of these twitches, and because of the all or none principle, although I'm not drawing this perfectly well, all the twitches should be roughly the same height. The muscle is generating the same amount of tension. If I have a muscle cell here pulling on this hook, and there's a spring there, the muscle cell will contract and relax, and contract and relax. And my body movement would be the equivalent of the amount of tension. That's a series of twitches. Now, if I continue to do so over a period of time, some of the muscle cells will stay with a little bit of tension in them. We basically essentially increase the tone of the muscle, and then the twitches are generating a little bit more tension. This is a concept called Treppie. The E is pronounced. I believe it's a German word for step, but I'm not exactly sure. You can look that up. But essentially, this is what athletes take advantage of when they are warming up. So rather than going out on a track cold and then trying to take off, you see sprinters and runners running, boxers shadow boxing, and athletes warming up. What they're doing is they're firing the muscle so much that our brain thinks, okay, listen, if I'm going to work this much, I need a few more motor units kind of contracting early on, increasing the tension so that I can get maximum force under my performance. And so it's a slight increase in tension due to a constant stimulus. Now, we still are getting twitches. Now, I'm going to erase most of this. I'll leave my original, actually I'm going to erase my original twitch too. Oops, sorry, both my erasers fell. I'm going to erase all this, and I'm going to draw a single twitch here. Now, what if I turn the dial to stimulate 10 times a second, and I hold that button down? Every time I stimulate the muscle, it's going to contract 10 times in a second. What I would see is this. I would start to see my first twitch because every time I shock it, I'm getting the same number of muscle cells contracting. And one of the things I want to draw next to this is a different concept. What if somebody is pulling on a rope, a single muscle cell or a small group of muscle cells, but the same ones, same amount of tension every time? They're pulling on a hook that's attached to a spring that's attached to a wall. So when I pull, the spring is going to want to shoot me back to where I started. You have to remember that the connective tissue coverings of the muscle cell, the fascicle, and the muscle itself, since it is connective tissue, has some elasticity to it. So now, when my muscle cell contracts, it's going to pull, and then when it stops contracting, the springs are going to pull it back to place. That's what happens in a twitch. Now, what happens if I tell the muscle cell to contract, and as it's relaxing, I tell it to contract again, and it pulls with 100% of its tension. As it relaxes, I tell it to contract again. So before it gets back to baseline, I add another twitch to that, and another twitch to that. I can see the tension in the muscle increasing by increasing the frequency, how often I stimulate. And I can see there's much more tension in the muscle than just a single twitch. We call that concept wave summation. Wave summation is nothing more than increasing the tension in a muscle by increasing the frequency of stimulation. Now, I'm going to take this a couple of steps further. Let's say I turn the dial up and I stimulate 100 times a minute. If I do it so rapidly, the muscle, and if I hold the button down long enough, the muscle will reach a point where it's maxed out all of its tension. The maximum amount of tension in a muscle is called tetanus. Whether it's one muscle cell or say 5 or 10 or 100 muscle cells, if every time I stimulate that muscle, the same number of muscle cells contract, due to the all or none principle, they always give me the same amount of tension. But if, when they contract, as they start to relax, I stimulate them again, then every time I'm increasing the tension of the muscle. Eventually there will become a point where I run out of room and the muscle cannot contract any harder. That's when we're at tetanus. There are two types of tetanus that we can witness in a muscle. This type of tetanus happens to be called incomplete tetanus. During incomplete tetanus, the muscle's locked at maximum tension with visible signs of relaxation. So essentially, I'm not stimulating so frequently that I've locked it out, but I've maxed it out and it's sitting here, it tries to relax, and I say, nope, keep contracting. Every time the muscle tries to relax, it's told to contract again. It's never allowed to start relaxing all the way back towards zero. Now, another concept associated with this that I've drawn here is this. We can also cause fatigue in a muscle. So a couple of definitions. Tone is the tension in a muscle while it's relaxed. A twitch is a single stimulus contraction relaxation phase. If I stimulate frequently enough, I can begin to add the amount of one twitch to another. We call that wave summation. The sum of 2 plus 2 is 4. I'm adding the twitches together to increase the tension. Maximum tension is called tetanus when the muscle's at peak tension. And excuse me, if I stimulate frequently enough, I can get to maximum tension with visible signs of relaxation. We call that incomplete tetanus. This is kind of what's happening when you're getting that last rep in and your muscles are shaking. Your brain's saying contract, the muscles say no, we want to relax, and the brain says no, contract, and they're kind of arguing it out. If you continue to do that, eventually the muscle runs out of ATP and can't contract anymore. That's what fatigue is. Fatigue is the inability of a muscle to contract due to a lack of energy, lack of ATP. All this muscles, all the myosin heads have burned up all the ATP and it can't continue to contract. It's out of energy. If you wait a little bit of time, as blood flow to the muscle continues, the oxygen and glucose will be converted by mitochondria into ATP, and then the muscle can perform again. If you don't wait long enough, it may not perform very long before you burn up all the oxygen again. So you have to wait in between reps or sets sometimes if you're working out. God, I'm so sorry I keep coughing. Please forgive me. Anyway, now, there's one more type of tetanus, and I think I can fit it in here before I run out of screen room. Let's say, actually, I'm just going to erase this incomplete tetanus, and I'm going to go to complete tetanus. I don't want to run out of space and waste some of the lecture. So, let's say I turn the dial up to, say, 1,000 times a second. I hold that button down. I'm going to stimulate so rapidly that the muscle almost instantaneously jumps to maximum tension and does not relax at all. I've completely eliminated the relaxation phase. I'm stimulating so frequently, this poor little muscle cell up here is going to pull and just be stuck pulling as hard as it can. That we call complete tetanus. Complete tetanus is when a muscle is stimulated so frequently that it is at maximum or peak tension with no visible signs of relaxation. During incomplete tetanus, we had visible signs of relaxation, but now we're stimulating so frequently, we've eliminated the relaxation phase, or we're locked at maximum tension with zero visible signs of relaxation. Eventually, the muscle fatigues. So now, we've gone over the two types of ways that muscles can perform, or at least how we increase the tension in a muscle. One of those is by increasing the number of active muscle cells or motor units called recruitment. And then the second way is increasing frequency of stimulation. Now, one last detail I want to give you, and then I'll stop this lecture and we'll move on to the last little bit here. I may try to finish it all in one video. We're getting close to the end, so let me finish this out. When it comes to recruitment and motor units, if you're following along in my notes, we're going to move on to we've done 57 and 58, we're moving on to page 59. We're going to talk about different types of motor units, different types of muscle. If I will have a very large muscle, for example, the muscles of the leg or the upper arm or the back, these muscles, their goal is to generate a great deal of power. I want to be able to throw a rock with a lot of power and hit a rabbit so that I can survive. Let's say if I were living in ancient times or living on my own, like, Naked and Afraid, the TV show. Great TV show to understand some human physiology, by the way. Or if I needed to kick something really, really hard, I kick a football a great distance. I have large, powerful muscles that are going to do that. Now, like I said before, if I have a lot of muscle cells, I can't have a single motor neuron for every muscle cell, or else my spinal cord would be this thick. The brain would be huge, and we just can't have that. So what happens is we have motor units. Now, in large muscles, if I have a single motor neuron, at the very end, that motor neuron can branch and go to many, 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 many muscle cells. Let's say every one of these circles represents a muscle cell. And then the telodendrium, blah, blah, blah. But nonetheless, I have all of these muscle cells. Now, I know I didn't draw a whole bunch, but let's say I have one motor neuron and I have 100 myofibers or muscle cells. Then that means every time one motor unit fires, I'm going to get a lot more power. If I have a small muscle, they don't generate a lot of power. What they do is what we call fine motor control. This would be what we call gross motor control. In gross motor control or gross motor movement, I have a lot of power. In fine motor control or movement, I have a lot of coordination, a lot of facility, very facile. And what happens is, because these muscles are small, a single motor neuron might only control 4 or 5 muscle cells, a small number of muscle cells. So one motor neuron, and I say 4 to 5, or only a few muscle cells or myofibers. If I do the math of myofibers to motor neurons, here I have 100 to 1, here I have, let's say, 4 to 1. If I do 100 to 1, what I have is considered to be a high ratio of myofibers to motor neurons. When I have a higher ratio of muscle cells per motor neuron, that means when one motor neuron fires, more muscle cells are going to contract. I'm going to generate a lot of force, but I'm not going to have a lot of control. When I want fine motor control for very small muscles, I can have a low ratio of myofibers to motor neurons. I have fewer muscle cells. I'm not going to generate as much power, but I can have very short, fine movements. And so if I took a real football here and I flicked it with my fingers, it ain't going to go anywhere. But if I flick it with my leg or with my arm, it'll move a great distance. Gross motor control